give the notes. Mycology is just simply the study of fungi, and of course, once we study fungi, we also look at the relationship of this fungi to other fungi and other organisms like bacteria, viruses, and all that. At the same time, it was also involves the study or the uniqueness of the biochemistry part of it, which sets them apart from other groups. What makes these fungi unique from other microorganisms? Alternatively, you can also as well say mycology is just a branch of microbiology which is concerned with the study of fungi that includes their genetic and biochemical properties. Example of this group of microorganisms are mushroom, yeast, molds, etc. This mycology dates back to around 18th century, where until we had thank you. So we're just saying that mycology is a branch of microbiology concerned with the study of fungi. Examples are mushroom, yeast, and molds. And we are also saying that until around 1830s, fungi was assumed to be a branch of plants, plant kingdom. But studies over time, where DNA and biochemical analysis was done, it later revealed that fungi are a separate lineage of eukaryotes, which they are multicellular, but they are different from animals and plants. And this was distinguished by the fact that the cell wall of this the fungi are made of chitin and glucans, which often surround the multinucleated cells. And this again was emphasized by a Greek philosopher who discovered that in 1830s there was some sort of a causative agent, a fungal causative agent that could cause what we call white muscardine disease of the silkworm. That is when studies started coming into being that indeed fungal is not just like any plant, but they have their own unique characteristic that made um, scientists to give it a unique classification. So in 1835, there was this scientist by the name Agostino Bassi, who was the first person to link an ailment with a fungal pathogen, that could cause disease even in humans. And he was able to demonstrate the first fungal agent by the name Baurelia bassiana to be a fungal agent that could cause what we call silkworm disease. Are we together up to there?
Yes.
Thank you. So that Agostino Bassi is among the very first scientists to give some evidence into terms of what we call fungal infection. So when the microscope was invented around mid 18th century, the microscope was able to reveal quite a number of distinct features that could separate plant and animal cells and also be able to show why is fungal quite unique, uh, not to be classified together with either of the two groups. And um, like let's say the other eukaryotes, their cell wall is usually made of cellulose, like in plants, But when the study was done for fungal cell wall, they found that it's made of what we call glucans and chitine or chitine, the way you pronounce it. So these two composition in the cell wall of the fungal was made the scientists to create a different kingdom apart from the animal and plant kingdom because of that differences. And of course, it was found to be malnucleated. Also, another study realized that fungal, as much as they are eukaryotic and heterotrophic microorganisms, they do not have chlorophyll like plants, and but can obtain its nutrients through what we call absorption of food or nutrients from dead or decaying organic matter in the environment. So again, that was a distinct characteristic that made, you know, the scientists to really classify viruses in a different category. Because if it's plants, they have chlorophyll and they can make its own food. But for the fungi, they cannot make its own food. Instead, they rely on what we call absorption of already decomposed food or nutrients from what we call dead, decaying organic matter. So that again, that's how the difference was also came in. Additionally, they also distinguished all other organisms, e.g., animals, by the principle of mode of vegetative growth and nutrient intake. So, according to the taxonomy of fungi or classification, this kingdom fungi has five major phyla that were established to not to based on what we call sexual reproduction or using what we call molecular data. Those two are the major characteristics that were used to classify fungi into five phyla. That is sexual reproduction and molecular data. So the five are as listed, what we call chytridiomycota, what we call chytrids. Number two is zygomycota, what we call conjugated fungi. We have ascomycota, sac-like fungi, Basidiomycota, that's club fungi, and glomerulomycota, the five classes. Are we together up to there?
Yes.
Okay. So as we proceed, we just want to look at essentially these fungi have two major characteristics. The yeast cell part of it and the molds. So when you talk of a yeast cell, it is as presented. It has those key characteristics, what we call the cytoplasm, the nucleus, Golgi apparatus, the lipid granule, the vacuole, membrane, and cell wall, where we also have mitochondria. So you realize that a normal cell have this kind of key organelles that assist the cell to, make its own food or even multiplication. So that is the typical bit of the cell wall. And you remember we said that the cell wall of this fungi is made of glucans and chitin as opposed to other compositions like cellulose for the plants. So when you look at some of the major characteristics of the fungi, we know that fungi are decomposers. Fungi can, just like bacteria, can be used to decompose what we call debris or materials that can be converted into useful. The cell of fungi differs from that of mammal in that they contain chitin and mannan, glucan, and glycoproteins. The mannan is found on the surface of the cell wall and it acts as an genetic determinant of fungal characteristics. So that is a key characteristic that fungi For the cell wall, the key component where it acts as what we call antigenic determinant is this mannan, part of the cell wall. So when you hear about the issue of, let's say, infection, being able to attach to a host cell and causing disease, the antigenic determinant into what we call a cell wall of a fungi is what we call mannan. And then glucan are kind of fibrils that are found on the cell wall that help in the strengthening it. So actually, this glucan is rather is used what we call giving the shape, giving the fragility, the what we call the the rigidness of the fungal morphology. So the fungal cell wall grow only in organic compound and they can only use carbon as a source of energy and are mostly found in decaying organic matter. So we are just continuing to characterize, to look at what defines the fungal, what makes us understand that fungi are different from other microorganisms or other animals and also different from plants. Yes, so um, again, in principle, there are two forms of reproduction displayed by fungal cell, that is sexual and asexual reproduction. Yeah, these fungi can reproduce in two ways as indicated. And when you talk of conidia, just simply produce during asexual reproduction, while spores are in what we call sexual reproduction. Okay, so as we go along, you realize that because of these two forms of fungal cell displaying the two ways of fungal reproduction, sexual and asexual, when you have conidial being produced, this that signify asexual reproduction, and when you have spores, then that signify what we call sexual reproduction. Again, there are two major forms or shapes of fungi. One, we call them yeasts. These are unicellular, simply meaning they occur in singles, in singular. They occur as unicellular, and therefore that means you cannot easily see them. They are minute subset of the fungi, and therefore you cannot see them. These yeasts they normally reproduce by budding, and after the budding, the daughter cells bud off, and the bud is known as plastoconidia. While the second form is the mold, this mold uh, develop into what we call the hyphae, and these hyphae are just simply like tube-like extensions. And when you have these several hyphae together, they form what we call a mycelium. When you have the hyphae together, they form a mycelium. These are under what we call molds. And for some of the fungi, the hyphae have segment, and the segment are what we call septa. Some hyphae of the fungi, which are tube-like, they're normally segmented, and with those segments we call them septa. And those which are not segmented, we call them asepta. Yes, and then we have what we call some species of the yeast cells, e.g., Candida albicans, they form what we call pseudohyphae, that's elongated, which are not simply means not true hyphae. Pseudo means not true. So pseudo hyphae, which are normally formed by Candida albicans, they are what we call um, not true hyphae. They also have a less rigid cell wall compared to one of the true hyphae. So number three, apart from the mold and the yeast, we also have what we call dimorphic fungi. These are cells that can grow into yeast or into mold form and can change from one form to another. Reason for being called dimorphic fungi. These are fungal cells that can grow, you know, can grow in yeast or in mold form and can change from one form to another. Dimorphic. At one point, they can be into what we call yeast cell, yeast form, and another time they can be in what we call mold form. But this, again, whichever form it's supposed to be, this depends on the environmental conditions. One of them is temperature. For this dimorphic fungi, at the body temperature, like if it's infecting you at that 7 degrees centigrade, they appear in yeast form. So mostly these dimorphic fungi, when they're affecting mankind at that temperature, that 7, you will diagnose them in yeast form. But when the temperature is lowered, they change into mold form. So most likely if the temperature is around maybe below 20, 25, 17, you will find them into mold form and most likely not in human body. Again, it also depends on the availability of nutrients because the yeast form requires more nutrients than the mold form. So where there are more nutrients, you will find the fungi in the yeast form. But if it's little, you will find them in what we call the mold form. So when you talk of classification of fungi, is based on mode of reproduction and micromorphology, which you talked about. Reproduction, you have what we call the perfect fungi. These perfect fungi reproduce both sexually and asexually. And of course, we talked about sexual, then spores, and asexual into what we call conidia. So, um, according to this, based on what we call the sexual spores, which are used to classify fungi into four categories based on sexual reproduction. We have what we call Ascomycetes, Basidiomycetes, Zygomycetes, and Oomycetes. These four classes are under the category of perfect fungi, classified based on what we call sexual reproduction, where it has spores. Then we have another category, what we call imperfect fungi. Remember these two classifications, perfect and imperfect, is based on reproduction. So these imperfect fungi, they are also called Dieteromycetes. These reproduce only asexually. No, they only uh, um, uh, reproduce asexually. That is one class by the name Deuteromycetes. So that is under mode of production. That is perfect fungi with four classes and the imperfect fungi with one. When you look at micromorphology, we have three. We talked about yeasts, molds, and dimorphic, which I have already talked about. So that is really trying to classify fungi into those kind of categories, classes, and all that. But studies have shown that most of fungi that affect or cause disease in human, they fall under what they call the heteromycetes group, which are under the class of imperfect fungi. Uh, that's that. Then we can also classify fungi based on what we call tissue of infection. The tissue or the body site that can be infected by the fungi can be used or can be the basis to classify fungi. For example, when you talk about superficial mycosis, we are simply talking about those fungi that can cause lesion on the skin and appendages. And this kind of fungi do not invade the living tissues. They don't go deeper in the tissues. But instead, this type of fungi are now called what we call dermatophytes. So when you talk of, for example, superficial mycosis, we know is a group of fungi that cause disease in terms of producing lesions or wounds or ulcers on the skin and appendages. They don't cause a deeper tissue infection. These are stomachoses. And under this, we have a group of fungi we are calling dermatophytes. So dermatophytes are a group of fungi that cause what we call superficial mycosis, or they cause what we call a infection of the skin or the superficial layer. These kind of microorganisms, the dermatophytes, do not affect living tissues. They only feed on what we call dead tissues, what we call the keratin, because they produce an enzyme keratinase that break down this keratin into what we call food form. And this keratin is dead tissues, dead skin. So the beautiful part of this superficial mycosis, they are not really pathogenic in terms of affecting or infecting what you call tissues. So under these dermatophytes or or superficial mycosis that cause superficial mycosis, we have three what we call genera of dermatophytes. We have what we call Microspora, epido, Epidophyton, then we have what we call Trichophyton. These three genera are under the category of what we call dermatophytes, and these are the one which causes superficial mycosis. They have what we call septic hyphae and microconidia. Septic simply means they have septate, and septate simply means segmented hyphae, and also microconidia. So these are what can be classified under what we call superficial mycosis. Then we have what we call subcutaneous fungi. These are a group of fungi that cause disease in the inner surface of the skin, and this can spread into the what we call lymphatic systems, and they can be introduced through what we call the skin down there. An example of this superficial, no, subcutaneous fungi are what we call Sporotrix and Sporotrix kengi, which causes sporotrichosis. And then we also have what we call Madunella mycetoma as an example of those fungi and what we call subcutaneous fungi that causes disease in that. Number three, we have what we call systemic fungi. When you hear of this, these are fungi that affect the internal organs, e.g., the lungs, kidney, etc., and are able to cause what we call severe diseases in human. So most of the fungi that cause severe life-threatening disease are in this category we are calling the systemic fungi, though some of them can cause what we call subclinical or no clinical manifestation. Somebody can have the disease, can have the infection of this systemic fungi, but will be unable to tell. if they are there. So from there, we have what we call, and an example of these systemic fungi are quite a number. We have Cryptococcus, Newformans, those are what we call pure yeasts. We have Histoplasma capsulatum, Dimorphic. We have Plastomyces, Dermatitis, what we call Dimorphic, and Coccidioids or Paracoccidioids, which are what we call dimorphic fungi. And then also we have this category of what we call opportunistic fungi. These are occasional pathogens that attack a patient depending on the prevailing condition, e.g., like what you call candida, that are termed as normal flora, but they can turn out to be, pathogenic when the immune system, is compromised. Um, so from there, that one just to show you, the yeast cell with quite a number of, those uh, so the composition of the yeast cell, yeast cell may attain, remain attached in the short chains, forming pseudomycelia, but they can. So the shape is that they're extremely variable in shape, being globose or oval, elongated or rectangular. Culturally, yeast cells are very polymorphic and are capable of assuming different forms in culture. And then, of course, the cell wall contains protein, lipids, and all that. So my main intention is this is a budding yeast. I thought I'd show you that whenever yeast is supposed to reproduce asexually, it just thins and then it buds off. So it's almost budding off to produce a daughter cell and of course the original cell. And obviously it will have the exact original composition of the original cell. From there, we look at the structure and the physiology of fungal molds. Molds consist of long branching filaments of cell wall of cell called hyphae. Molds contain long branching filaments called hyphae or singular hypha, and a tangled mass of hyphae visible to the unaided eye is mycelium. That is singular, but when we talk of many are mycelia, this is a mass of more than one hyphae together. But in some molds, cytoplasm passes through and among cells of the hyphae and interrupted by the cell wall. Those which are, of course, cross walls are called septate fungi, that is septa. Then those which shift from yeast form to mold, vice versa, dimorphic and all that, that one we've already covered. Then when we talk of the structure of the physiology of one of this, sorry, and that's to key thing. That's an example of the mold form. We have what we call the aerial hypha. The hypha, which looks like roots on the down there, subsurface hypha, so to speak. Then when it grow up, at the end there we have what we call conidiospores. So conidiospores, once they bud off, you see the budding, when they bud off, they germinate through forming long tubes like, then from there again they form. So that's how the mycelia and the hyphae is formed, the mold form. Then of course I showed you the yeast. So an example is like the bread mold, the mold which is found on the bread. When you have it, this is how it looks like. It has those rhizoids down there. We have the stolon. Then once the sprongiophore has enough mature molds, it will open up the spongium will open up so that the spores, are released. And then here, another type of bread mold, what we call the Rhizopus stolofina. That's how it appears. So this is just to show you when you talk about mycelium, the mycelia, the hyphae, how they look like, how does it happen, and obviously once the spongium bursts or open, many and millions of the spores are found. Yes. Yes, that's now the end of our introductory bit of the fungi. That's the introductory bit of the fungi. So I want us to take some bit of break, and then we want now, when we come back, we want to look at the pathogenesis now. We look at those fungi that causes disease. We were just laying a foundation to understand what is mycology, when was it discovered, what are these the fungi, the classification. We've talked about like the perfect fungi, the imperfect. We've talked about those superficial mycoses, subcutaneous. We've looked at systemic and examples. Now that we want to internalize that, on a later time, I want us to now look at some of the key diseases that we can acquire from these fungi.